In gene interactions 1, I had talked about the what you call changes in the monohybrid ratio. Okay, so where there would be change of the monohybrid phenotypic ratio of 3:1 or 1:2:1 genotypic ratio. Now in gene interactions 2, I will be talking about some changes of the dihybrid ratio. The changes of the dihybrid ratio. That means in dihybrid ratio of Mendel, he has said that 9:3:3:1. But now here we will talk about some interactions wherein we will find that the ratio is different, like 9:7 or 15:1. So such interactions we'll be talking in the second part. Next, let us come to this another one, which is called duplicate gene action. So in duplicate gene action, what happens? Suppose in this case, this is an example of the floating and non-floating habit of rice. Okay, this example which we are giving is floating and non-floating habit of rice. So if you put rice grains, floating habit of rice. So if you put rice grains in water, that can either sink or float in water, and accordingly this genetic composition. So here you see all are dominant. That means two genes are there, capital W1 and W2. These are there, and then small w1, small w2, and small w2. So this dominant, so this is non-floating, it will cause the rice to sink, and here in this case all recessive, and so it will cause the rice to float in water. Now here we know to find out the gametes, okay, and then these two will fuse, correct? These two fuse and you get a F1, okay. Next what you do an F1 cross. Next you do a F1 cross, in which what you do, say capital W1, small w1, capital W2 and small w2 cross capital W1, small w1, capital W2 and small w2. Correct. So when you do this cross, you find out the gametes. So these are the gametes which you have found out. These are the gametes which you have found out. Correct. 4 gametes which you have found out, and here also, again the other side also, you will get the same gametes. Correct. Okay. So you have to just write the chromosomes like that, and you write it like this. Now W1. So here this will be small w1, then w2, and this will be small w2. So your gametes will be w1, w2, 1, w1 and small w2, small w1 and capital w2, and small w1, small w2. This is how you find out the gametes. Anyway, after finding out the gametes, you do the cross like that. This and this, it will go, and you get it here. Similarly, this and this, and you get it here. Correct. I think this already discussed these things, how you do it. Yes, this and this you get like that. Similar w1 w2 cross small w1 w2, so capital w1, small w1, capital w2, small w2. You get it in this manner, this square. After that, now how to find out the characters? What will be the character here? So according to duplicate gene action, what they are saying is that even one dominant gene, even one capital W is enough for the non-floating habit. Even one capital W is enough for the non-floating habit. Others are all duplicating. That means if two capital W will be there, then also non-floating. If three capital W's are there, then also non-floating. And if capital W4 are there, then also it will be non-floating. Only in this case, this will be floating. Correct. If this is the case, then you will get only floating here. Correct. And all others will be non-floating. So what is the ratio that you get for duplicate gene action? So the ratio which you get for duplicate gene action, since this is 16, so it will be 15:1. Okay. So this is what you have to read it here. I am not reading it here for the time purpose. You read it, and this is what they have said, that even a single dominant gene is enough to express the character, and for the other character to be expressed, all recessive genes has to be present. Okay, so this is what we call as duplicate gene action. So such ratios we didn't find in Mendel. Mendel had the dihybrid ratio of what? 9:3:3:1. But here you are getting a ratio or interaction which is 15:1. Duplicate gene action. Next is complementary gene action. So here again, we are taking an example of flower color in sweet pea. So here we are taking these plants, say purple, capital C, capital C, capital R, and small c, smaller. So purple, and then you find out the gametes, capital C and capital R, here small cr, and then this is what we get, the F1, correct? This is the F1. Now we do a F1 cross, yes, so we will get the F1 gametes. I have told you how to find out the gametes. You do it, F1 gametes. So here capital C, you put it here small c, capital R, and you put small r, and you find capital C, capital R. This will be one combination. Capital C will take small r, second combination. Small c will take capital R, third combination, and small c, smaller. So this is what we have put it in the gametes, correct? This is the gametes which you have put, and similarly for the other parent also same thing. So again male and female gametes we have put, correct? Next, here you have to remember that again what you call, here same crossing we will do like this, capital C, capital C, capital R, the same process, capital C, capital C, capital R, smaller. So we continue like this and we get this Punnett square. But after getting the Punnett square, the main thing would be that how you find out the ratio, correct? Now for that, here what they've said, the production of one of the two phenotypes of a trait requires the presence of dominant alleles of both the genes controlling the concerned trait. When any one of the two or both the genes are present in the homozygous recessive state, the contrasting phenotype is produced. Okay. So here then what can happen? That means here for a character to be expressed, both the dominant genes are required. So capital C and capital R. If both capital C and capital R is there, both the dominant genes, then only you would get a character, say purple. Okay. You will get a character purple. But then if any one of them is there, then what you will get? According to that interaction, what will you get? If any one of them is there, then here what they've said, the production of the two phenotypes of the trait require the presence of dominant alleles of both the genes. Yes, dominant alleles of both the genes. But if any one of them is there, so any one means if say capital C is there and capital C, but here smaller is there, then what will be the character? The character will be white because it doesn't have any expression. Both the dominant genes has to be present. Correct. At least one capital C and one capital R has to be present. Similarly, if suppose small c and here capital R, then also it will be white. Have you understood? So that means both the dominant genes has to be present for the character to be expressed. So both the dominant genes here, capital C, capital R is there. So this will be purple. Here also C and R, capital R, there purple. Here purple. Yeah, in this case, what do you see? Here capital C is there, but small r is there. So another dominant is not there to complement it or help it. As a result, this will be what? This will be white. This will be again purple. This will be white. This will be purple. This will be purple. Yes, but this will be white. Even though these two dominant genes are there, another dominant gene is not there. They're all recessive genes, isn't it? And here this will be again what you call small c is there, correct? So here also it will be white. So this will be white, this will be white, this will be purple, this will be purple. So like this, what you get then? So you will get that 4, no, 6, 7. Oh, sorry. Here this will be what? This will be white, correct? 7, 8, 9. So here 1, 2, 3, 4, 5, 6, 7, 8, and 9. So there will be 9 purple. 9 purple is to 1 white. 9 purple is to 7 white. Okay. So here the ratio will be 9 is to 7. So this you have to remember, 9 is to 7. So you read the statement again if you don't understand, and you will get it what it will be. Next comes the supplementary gene interaction. In supplementary gene interaction, what happens again? You do the same here. Okay, you do the cross for supplementary gene action, and here I am giving you this example is for what you call supplementary gene interaction. This is also called as recessive epistasis. So here again you do the cross capital R, capital R, PP, and small r, small pp. Then when you do the cross, you find out the gametes. You then find out the F1, yes, and you do the F1 cross. You find out the gametes. I think now you can find out the gametes, and you get these things. Here what they are saying, the dominant allele of one of the two genes governing a character produces a phenotypic effect of its own. But when it is present with the dominant allele of the first gene, it modifies the phenotypic effect produced by the gene. So suppose capital R has its own expression. Suppose capital R has its own expression. Suppose that expression is red. This is what they are saying. But when with capital R, capital P is there, P is modifying capital R, so that becomes what? It becomes purple. Okay, so P is modifying it. That you have to remember. And but if only capital P is there and here smaller is there any expression for P? There is no expression for P. So as a result, this will be what? This will be white. Okay, so that means wherever capital R only is there with small p, with small p, with small p, then it will be red. So in that way you find out the ratios, and you will find what the ratio to be 9:3:4. You do it yourself and see whether you understand or not. 9:3:4, where 9 will be what? You can check R and P. So 9 will be purple, yes, and red, which will be red. Suppose I can give you one red. Suppose this is a red, yes. Then this is small p, small p. So any like that. This also will be red, yes. Then this will be red. Okay, red. What will be this? This will be white. This will be white. This will be white. This will be white. So purple, red will be 3, and white will be, and others are all purple. Okay, so here this is what they are saying, that dominant allele of the two genes governing produces a phenotypic effect of its own, but when it is present with the dominant allele of the first gene, it modifies the phenotypic effect produced by the gene. So here capital P is modifying capital R. Okay, this is okay. And why we call it as recessive epistasis? You can see here, even though a dominant gene is present, a character of recessive gene is being expressed. So it is recessive epistasis. So even though dominant gene is present here, capital P is dominant, but recessive character is being expressed. So this is what we call as also recessive epistasis. Okay, so this much in the first part of gene interaction. Thank you.